Welcome back from the break. We're on the home stretch now. Moving into our first presentation in this session, FDA describes the role of the Structured Data File and Computational Toxicology Consultation Service QSAR review workflow, which includes structure verification, generation of model predictions, and databasing of results. Our presenter, Dr. Naomi Kruhlak, has worked for CDER as a computational toxicologist for over 20 years, developing and applying quantitative structure-activity relationships, also known as QSAR, models to support the regulatory review of pharmaceuticals. She is the scientific lead for CDER's Computational Toxicology Consultation Service and is the principal investigator on 3 FDA CDER research collaboration agreements with commercial QSAR software vendors, as well as an interagency agreement with NIH generating in silico-based drug safety predictions. Our next presentation will describe what substance information is captured and communicated by the GSRS team. Our presenter, Dr. Frank Switzer, is a member of the Health Informatics staff in the Office of Data, Analytics, and Research in FDA's Office of the Commissioner. Dr. Switzer has been the primary data manager for FDA's Global Substance Registration System since joining FDA in 2008. Our final presentation will cover the history, design approach, current features, and potential future uses of the Global Substance Registration System. Our presenter, Tyler Perea, is a informatician in the Health Informatics staff, Office of Data Analytics and Research, Office of the Commissioner. He supports the GSRS project, which coordinates with all the FDA centers to link common data together in a standardized open source framework. He has over 15 years of experience in software development, scientific data management, and general data science. Please join me in welcoming our first presenter, Dr. Kruhlak.
Thank you for the introduction. My presentation will now focus on how we can improve the review of quantitative structure-activity relationship data through the use of structure data files, or SD files. This is an outline of my presentation. It has 2 parts. The first part will cover the basics of QSAR modeling, how a QSAR model is constructed and how it's applied. I'll talk about the types of chemical structure formats that are used to not only construct a model but to generate a prediction using a QSAR model. And then I'll talk specifically about the benefits of one of those formats, the structure data or SD file format. And then I'll move into how that is used under the ICH M7 harmonized regulatory guideline, specifically how QSAR is applied in that context, the importance of structural accuracy when generating predictions and how that can be facilitated using an SD file. And also I'd like to highlight the importance of using structure-linked databasing to support a QSAR analysis workflow. So let's begin with the basics of QSAR. So what is QSAR modeling? QSAR stands for Quantitative Structure-Activity Relationship, and it's a type of modeling that identifies associations between attributes of chemical structures and biological activity. And in the context of this presentation, we're talking about mutagenicity. It works under the general assumption that similar molecules exhibit similar chemical and biological properties, and therefore that toxicity can be explained by chemical structure. The modeling process itself learns from the results of actual laboratory testing. So when we're talking about generating a prediction for mutagenicity, we're modeling data that comes from the Ames assay, which is the gold standard for assessing mutagenicity. We would use a computer to examine a large number of chemical structures, each with their associated activity score in the Ames assay, to define certain pieces or substructures that are statistically associated with a positive response. And those pieces will then be set aside and labeled as structural alerts. But we take it a step further to identify other attributes of structure that, when present along with a structural alert, can actually mitigate activity, can lead to an absence of a positive result. That information is then extracted from the training set and stored as a model, and then we can use that model to make a prediction of a new chemical's toxicity based solely on its chemical structure by checking for those structural alerts and also mitigating features. And of course, the benefit of this type of an approach is we can fill data gaps when empirical or experimental data are unavailable or inadequate. And to clarify this definition going forward, QSAR is a quantitative relationship that's typically seen in statistical-based models, so those are models constructed through machine learning, whereas SAR is a qualitative relationship that's most often coded into expert rule-based or human-derived models. But collectively, when we refer to both of them, we put the Q in parentheses and we write it as QSAR, as shown in red on the right-hand side. So there are three things that go into building a QSAR model. First of all, we need to have a set of chemical structures that are represented in a format that's interpretable by a computer. And we do that through the use of what we call molecular descriptors, which essentially serve as a digital fingerprint for each of those chemical structures. And then for each chemical structure, we need to have some numerical representation of the activity of that chemical at the endpoint that we're constructing a model for. So in this case, we're building a model to predict bacterial mutagenicity with the Ames assay. And so each of those chemical structures will have an assigned score that denotes either the compound being positive, so it will get a 1, or negative, where it will get a 0. We then need to bring in a computer algorithm or some other kind of method for extracting the patterns between the chemical structural features and their activity to pull out those structural alerts and also those mitigating features that will define the model itself. So once that's all pulled out using our algorithm, that information is stored as the QSAR model. And we now come along with our new compound for which we would like to generate a prediction. This is typically something we don't have experimental data for already. That structure needs to be encoded in a computer-readable format. You'll see that as a theme throughout my presentation. And the model will check for features within that structure to determine whether any structural alerts are present or whether any mitigating features are present. And based on the relative weight of those features, it will then generate a prediction. And in this case, the model considers the weight of those mitigating features to outweigh the presence of the structural alert, supporting an overall negative prediction. There are a number of ways we can represent that chemical structure in a computer-readable fashion. Of course, we can represent our structure in 2 dimensions or actually in 3 dimensions. The majority of the QSAR models that are used for impurity qualification are 2-dimensional models. The file formats that most QSAR software use are shown here. And a common format is the SMILES code, which is a linear string of characters where each letter denotes a different atom. Parentheses denote branch points. We can see an equal sign being used as a double bond. The INCHI code is a more recent standard that's used for representing chemical structure formats where each section of that code refers to a different type of chemical attribute. So there may be a section for isotopes or a section for stereochemistry. And then probably the most common format is actually the mol file or the SD file, where the mol file represents a single chemical structure, but an SD file can allow us to end-to-end join these individual structures to create one file with multiple structures and their associated data in a single place. And just to show you what that mol file or SD file would look like if you opened it up as a, in a WordPad or something similar, word processing application, we can see that it actually has a connectivity table that indicates which atoms are connected to which other atoms and also the coordinates for those individual atoms. So what are the benefits of the SD file? Well, as I mentioned, it allows us to actually put multiple structures into a single file, but separated, and they can each have associated data. So this has the benefit of allowing us to batch process tens to hundreds to even thousands of structures if it were necessary. This is helpful because they're all in one place, and we can actually input an SD file into QSAR modeling software and generate predictions for tens or hundreds of structures in one go. The SD file is also an open standard, so it's not related to a particular commercial software, so it can be opened in a lot of freely available tools and manipulated. It has a small file size. It's based on ASCII text, as I showed in that previous slide, and so it makes data transfer facile. So this format really works well for regulatory submissions of drug impurity structures under ICH M7. And according to our quick guide that has been presented by Marlene Kim in this workshop, there are a number of associated data fields that we would expect to see for any chemical structure that's being sent to the agency in an SD file. And those fields include name, application number, ID, role, Unicode, CAS, or notes. So now let's move into QSAR analysis under M7 and how we would use an SD file in that context. So just to refresh, ICH M7 was published in June of 2014 and revised to R1 in March of 2017. It's now under another revision to R2. It focuses on the assessment and control of DNA reactive or mutagenic impurities in pharmaceuticals to limit potential carcinogenic risk, and it describes how a hazard assessment should be conducted on a pharmaceutical impurity to classify it as mutagenic or non-mutagenic. This is done based on experimental Ames data, if available, but in the absence of that data, a QSAR model, or actually two QSAR model predictions, can be used to support that qualification. The QSAR models that are used for this purpose should be predictive of the outcome of the Ames assay, so they need to be trained on data from chemicals that have already been tested in the Ames assay. And two complementary modeling methodologies should be used. One of the models should be statistical based, so that's our machine learned model, and the other one should be expert rule based, so that's our human expert rule derived model. The models need to be adequately validated and documented according to the OECD's validation principles to demonstrate that they're fit for purpose. And once those models are applied, if no structural alerts are identified in the structure of the impurity, then no further genotox testing is required at that point. The guideline also includes an important provision for the application of expert knowledge, and this is where we can bring to bear additional information or supportive evidence to help us understand the relevance of any positive, negative, conflicting, or inconclusive predictions to support our final conclusion. So types of analysis that would fall under this umbrella of expert knowledge would include, firstly, identifying and interpreting the alerting portion of the molecule, as well as any other mitigating features that may have been identified, and to think about those in the context of mechanism of reactivity. So if we look to the left at the structure that I showed on one of my previous slides, we can see the model identified not only a structural alert corresponding to the aromatic amine, but two mitigating features which are substituents in the ortho position. And in the context of mechanism of reactivity, these make sense. The aromatic amine has the potential in some cases to be metabolically activated to form a nitrenium ion that can then go on to form an adduct. However, if we have substituents on either side of that on the ring, it's likely to limit its ability to be metabolically activated and go on to form an adduct. So it makes sense that the model would consider these features and yield an overall negative prediction. We also have the ability to look at the underlying training set structures that support the identification of the structural alert and also mitigating features to make sure they're structurally relevant to our impurity of interest. And in cases where we may consider that the model actually doesn't have enough information to support a prediction, we can bring in information from other supplemental sources to help us arrive at a robust conclusion, a positive or negative prediction. This application of expert knowledge is something that we routinely do to all QSAR analyses conducted internally at FDA CDER because we believe that it improves the overall confidence and accuracy of predictions. When the agency receives QSAR predictions from industry, they are initially triaged by DMF review staff. Any predictions that are straightforward and are adequately documented will be accepted as is, but any predictions that are questionable or the conclusions are not well supported, or perhaps the models that were used to generate the predictions are outdated, then a consultation request will be sent forward to my team for an internal QSAR analysis. And for us to be able to do that QSAR analysis, we need to have the structure in an electronically readable format. So if we don't receive that electronic format from the industry submission, we need to create it ourselves. And we have to create it not only for the impurity we're running the analysis on, but also its corresponding API structure as a comparator, because we will run a prediction on that as well. And we will create that electronic format from the submitted image. The problem there is that the quality of submitted images varies widely, and in some cases it's not entirely clear what makes up the structure. This is an example where one part of the structure has become distorted, and it takes additional time for us to figure out what that should really look like. So we have a final structure that we know is accurate to then run our QSAR analysis on. So this redrawing steps takes extra time, and it also has the problem that it can introduce errors, and that's something we really want to avoid. And since the prediction that we're going to be generating with our model is based on the chemical structure, we absolutely have to get that structure right. We can't have any errors in it. An incorrect structure will lead to an invalid prediction. And in fact, even slight changes in structures can lead to quite substantial differences in predictions. For example, if we look at these two structures here, we can see that the top structure differs by the presence of a methylene carbon between the ring and the amine compared to the lower example. And of course, that means that if we don't have that methylene group present, we now have a known structural alert. We have an aromatic amine moiety that is likely to be mutagenic, whereas if we draw the methylene carbon in the correct place, we have a molecule that's very unlikely to be mutagenic. So that minor change makes a huge difference. Another example would just be positional isomers. If substituents on a ring are not put in the correct positions, this can also change the predictions that we get from our models. And so using the amine as another example, if we have T-butyl groups immediately around that amine in the ortho position, that extra steric hindrance is going to lead to a lack of mutagenic potential. However, if we move that T-butyl group to the meta position, that actually is going to change the outcome of some of the models that we're using. So for that reason, if we can have the structures already in an electronic computer readable format when they come to the agency, if we need to do any follow-up analysis, we already have that accurate structure on hand, and that improves our review accuracy and efficiency. It's recognized that there is some pre-processing that's needed for QSAR models. If we receive a structure in an SD file, we do need to sometimes make some minor changes to allow it to be technically interpreted properly by the QSAR analysis, but these really are only minimal. So the types of minimal changes that we might need to make to a structure that we receive would be removing things like salts and counterions or some charges to create a structure that is then appropriate for QSAR processing, like in the example I have on the right-hand side where the sodiums have been removed and then also the oxygens have been protonated. Another example of minimal processing that is sometimes needed is for certain types of functional groups, such as a nitro group, where we want to draw that structure with a charge-separated nitrogen-oxygen bond rather than a pentavalent nitrogen. And that's just because of the way that the training set structures are drawn in the models. So chemical structure is really important, and computer readable chemical structures are really important, not only for the QSAR analysis, but also for answering other key questions that may come up during our review where we need to refer to a database of information. And these are some of the typical questions that we are often answering when we are looking at an impurity structure that's been received by the agency. We want to know, has the agency reviewed this particular impurity before? Are there experimental data available for this compound already? There's often public information that's available, toxicology data for this impurity that would be useful in allowing us to qualify it. In the absence of experimental data, have we previously performed a QSAR analysis for this exact compound? And furthermore, are there data for structurally similar compounds that might support our expert review? The most straightforward and accurate way for us to address these questions is to use a structure-based query. But in order to do that, we need to search our database using a computer-readable structure. So having that structure already in that electronically readable format in an SD file really facilitates that review, that ability to search our databases. So databasing is essential for QSAR review. The Computational Toxicology Consultation Service, that's my team, maintains an internal database of about 35,000 chemical structures linked to published toxicology data and also past QSAR analysis reports that we've generated internally. And new structures that we receive can be imported directly into that database using the SD file format, and that improves our efficiency and allows us to track those structures going forward. Reviewers at the agency have the ability to search the database for past internal QSAR analyses, and that's helpful because they can see if we've already reviewed a structure and run QSAR and issued a conclusion on it, and that eliminates the need for duplicate consultation requests for the same compound. And furthermore, our QSAR analysis staff can use the database to support their expert review of QSAR predictions by enabling searching to be done of the database using structure-based queries. So that can be done using exact searches or substructure searches or global similarity searches, all of which allow them to find relevant analogs to support their evaluation. Furthermore, they can also rapidly retrieve and review past regulatory decisions for related compounds, and that ensures consistency across all the structures that we evaluate. So in summary, QSAR models make predictions of toxicity based on chemical structure, and this requires the structure to be in a computer-readable format. The SD file format is commonly used in Cheminformatics, and it encodes multiple chemical structure information and associated data in a computer-readable format. It's also highly suitable for regulatory submissions of drug impurity structures under ICHM 7. In a QSAR review workflow, SD files eliminate the need for us to redraw structures, which puts us at risk of introducing structural errors, so we can eliminate that step. And it facilitates the application of expert knowledge to QSAR predictions by enabling structure-based searching, because we already have the structure in that computer-readable format. It also enables direct databasing of incoming impurity structures for future tracking purposes. So in short, drug impurity structures submitted to the agency in the SD file format improves our efficiency and also our accuracy of QSAR analysis review. And with that, I would like to acknowledge members of the CTCS team, as well as other FDA collaborators in CDER, as well as in the Office of the Commissioner, and our external collaborators, LeadScope, LASA Limited, and Multicase for providing the QSAR software that we use internally. Thank you for your attention.
Good afternoon. My name is Frank Switzer. I'm a chemist in the FDA's Office of the Commissioner in the Office of Data Analytics and Research. Among other things, our team manages FDA's Substance Registration System, which generates unique ingredient identifiers, or UNIIs, that are used throughout the regulatory process. The views you will hear today are mine. They do not necessarily reflect the views of the FDA. FDA's Substance Registration System is now a worldwide effort. The global SRS has been developed primarily by the Informatics team at the National Center for Advancing Translational Sciences at the National Institutes of Health. This slide shows the current public GSRS homepage. The images reflect the breadth of coverage in the system, that is, everything from an atom to an organism. This open source software has cheminformatic, or chemical structure, and bioinformatic, or sequence search tools. It is populated with FDA public substance data for public use. It was designed to facilitate global monitoring of human and animal, medicinal, food, tobacco, and cosmetic products. GSRS provides unique substance identifiers consistent with the ISO 11238 standard originally published in 2012. My colleague Larry Callahan and I were the editors of that standard. So why did we create GSRS? Regulators need a system that can accurately track the global supply chain of ingredients. These ingredient substances are increasingly macromolecules such as monoclonal antibodies or nucleic acid vaccines, modified cells, or biological extracts. This GSRS common system with common content was developed to assist the global community in managing the diversity of substance information that regulators need. FDA's GSRS generates unique ingredient identifiers, or UNIIs, used in electronic product listing. At present, about 144,000 U.S. marketed products are listed on the Daily Med website published by the National Library of Medicine. All of these listings must use UNIIs to identify their ingredients. UNIIs are also used for other regulatory activities, including both investigational and marketed products. This slide represents the ongoing international harmonization work that primarily involves the U.S., Europe, and the World Health Organization. The ISO IDMP, or Identification of Medicinal Products, referenced in the slide, refers to a set of 5 standards created to manage information around medicinal products. GSRS is central to these harmonization efforts. It was built to implement the ISO 11238 substances standard, which is a part of this suite of standards. The original intent of IDMP was to create an international drug dictionary. That goal has not yet been fully realized. The drug world is a sea of terminology. This slide shows one example. Substance names are good for tracking, but there are a lot of names that change based upon development phase and jurisdiction. Here we have an investigation or company drug code, multiple generic names, multiple brand names, and chemical names, all describing the same drug substance. GSRS provides a type of Rosetta Stone for managing all this complexity. So how is the GSRS sausage made? We first choose the substance type that determines the type of identity description that will be used. We then follow a set of rules to create a description or definition of that substance. A second expert then reviews this substance identity description to confirm accuracy and uniqueness and generates a UNII. If the substance information is private, this GSRS record is private. If the substance information is public, that record is published. Many GSRS records begin as private, but are later become public. Using a single UNI for a substance allows FDA to track that substance from investigational through to marketed product development stages. GSRS has 5 substance types: chemical, polymer, protein, nucleic acid, and structure diverse. Mixtures are described as multiple instances of these 5 types. Chemical substances are described by sets of atoms and bonds commonly referred to as chemical structures. Polymers contain structural repeat units. Proteins are primarily described as a sequence of amino acids connected by amide bonds. Nucleic acids are similarly described as a sequence of nucleotides. Structurally diverse substances are primarily described by organism taxonomy and parts or fractions of the organism. The example here is Echinacea purpurea or coneflower. Chemical structures need to be fully described in GSRS, including their stereochemistry. 2 common examples are shown below. Cholesterol has 8 stereocenters, while tetracycline has 5. Configurations of stereocenters are typically represented through dashes and wedges on flat structures. Unis for at least 20,000 substances may be found in Wikipedia info boxes. This slide has a link to the UNI Wikipedia page. UNIs are always 10 alphanumeric characters. They cover the full spectrum of ingredients, and they are free to use. Most UNIs users first consult the UNI search on Precision FDA. This site is updated about once a month, and the full data set is downloadable as a pair of text files. It currently has about 135,000 UNIs with over 1 million associated names and codes. Users often search by chemical abstract service or CAS registry number. The site currently gets about 2 million hits per month, and the vast majority of these hits are direct URL queries for the UNI, as shown in the second bullet. If a user does not find their UNI on that site, they're encouraged to request a UNI through this mailbox that I manage. If the substance is a chemical, our team needs information that leads to a chemical structure. A CAS registry number is often sufficient. More complex substances like polymers need additional information. Safety and technical data sheets are often good starting points for that research. They often include brand names that lead to more precise identities. Precision FDA also hosts a GSRS instance where users can securely register and share their own substances. They can also create customized reports of the GSRS information. Significant effort went into setting up this tool, but no one has used it yet. Our team is very willing to provide training for anyone who's interested. This slide shows some of the information available in the UNI search for dalfampridine. Included are the molecular formula and the INCHI key, which is a text string that represents the chemical structure. There are also several link outs to other resources, including the full GSRS public record at NCATS and listed products in DailyMed. This slide shows some of the information in GSRS for dalfampridine. The link to the site is on the right. Here's an example of dalfampridine linking to DailyMed through its UNI. GSRS identity descriptions can get complicated. Examples of this are sugars, dyes, and peptides. The next 2 slides show the example of the monosaccharide galactose. There are multiple common ways to represent this molecule. They include Fisher projections, Haworth projections, cyclic, and linear acyclic structures. For GSRS, the preferred description is the acyclic drawing on the left. When representations become complicated, GSRS has the ability to create alternative definitions. In the case of galactose, there are multiple forms in rapid equilibrium in solution. This alternative definition attempts to capture the equilibrium between cyclic and acyclic forms. Alternative definitions are particularly important for peptides that can be represented both as chemical structures and as proteins. GSRS currently has about 1,200 of these alternative definitions. Behind the FDA firewall, GSRS connects to US and Canadian products, applications and adverse events from multiple FDA centers, and US and European clinical trials. Substances have become the linchpin of FDA's data management. I would be remiss if I didn't say thank you to the many people who continue to move this work forward. Marlene and Tyler are presenters in this session, while Larry expands the influence of GSRS on the Blue Planet. Thank you for your attention. We will be answering questions at the end of the session.
Thank you, Frank, for that introduction to GSRS and UNIIs. I think it's a good backdrop for this following presentation. My name is Tyler Perrier. I'm a chemist at the Health Informatics staff group. I'm part of the Office Commissioner, ODT, and ODAR. My topic today is on machine-readable synthetic pathways in GSRS and KASA. So first, a disclaimer. The mention of commercial products, their sources, their use in connection with material reported herein is not to be construed as either an actual or implied endorsement of such products by the Department of Health and Human Services. This presentation reflects the views of the author and should not be construed to represent the Food and Drug Administration's views or policies. Okay, so today we're going to be talking about the Group 4 Specified Substance Manufacturing Form, the G4SSM form, in FDA's Global Substance Registration System. That's a lot to say, and it's a very technical term. In practice, what we really mean is we're going to be talking about structuring synthetic pathways using elements that are present in the ISO 11238 standard and using the GSRS implementation of that standard specifically for the Group 4 manufacturing information. Just a few terminology notes. Throughout this presentation, I am mostly going to be using the term synthetic pathways to describe the data in question. There are other terms people use for this that are sometimes more common: synthetic route, synthetic diagram, flow diagram, just synthesis diagram. In this case, there are subtle distinctions between each of those, I'm sure, but in practice, we will use those terms somewhat interchangeably, but we'll mostly be using the term synthetic pathway to describe the data in question. So what is a synthetic pathway, and what is in a synthetic pathway? Well, what you see on the screen is a simple example of what we consider to be a synthetic pathway and the kinds of information that we would want in a Group 4 specified substance manufacturing form. We really want the set of chemical reactions and the set of full processes that take some input material and chemically transform it into a resulting material, which may be an intermediate substance, but it may also be an API. As you can see on the screen, along the way, you're starting with some input material, some starting material, and you go through a set of chemical reactions that transform that material chemically, adding functional groups, removing them, adding protecting groups, forming salts, things of that nature. So it is a set of reactions that have individual reaction steps that result in a final material. So these synthetic pathway diagrams are quite useful to the agency, and they're quite useful to chemists in general. So it's worth exploring a little bit of how we get them now. Currently, these very informative pictorial diagrams are submitted to the agency attached in a PDF. So a digital file that really is just an image of this scheme, this synthetic pathway, submitted as part of the ECDD submission pipeline. For a chemist, it's very useful to have this in a nice, readable digital format, in a similar way you might find in a journal article or publication. Chemists can look at these kind of diagrams quickly and understand the general techniques that are used, and they can also often quickly identify the kinds of areas where there might be side reactions and the kinds of areas where there might be issues. These synthetic pathways are extremely useful to reviewers and just useful to chemists in general. And they're useful enough, in fact, that ICH ACDD standards specifically call them out as important submission elements in the following sections shown on the screen. In the ICH documents, they refer to them as flow diagrams, but they refer to the same thing, these synthetic pathways, the sets of reactions, chemical reactions that are used to produce the material. Now, this is extremely useful for the agency for a variety of reasons, but for one, it's very useful to have this information in so that we can monitor things that may be potential impurities, potential side reactions, potential contaminants. It's very useful for us to know how these things are made and to be able to anticipate potential problems. It's also especially useful more and more as we get into a global economy, because we need to be able to monitor issues that arise from a global supply chain. We're not just interested in monitoring the supply of the final drug products. We're also interested in monitoring the supply chain for the components that are useful in making those final drug products. And to do that, we need all of this data entered in a structured way to the agency. So some background on ISO 11238. Frank went into this a little bit, so I won't spend too long on this. But the ISO standard for substances gives a framework for defining materials. And at the core of that framework is the substance level information. Those are the things, the materials that are defined uniquely, chemically, based on their immutable properties, and they get a unique identifier regardless of purity and grade and form. That is the core of what ISO 11238 is about. But there are other layers that give more specified versions of those substances that are appropriately called specified substances. There are 4 groups of these specified substances. I won't go into what each of these are in detail, but just know that they can capture information at a more granular level than just the substance level would, but they always refer back to those substances, to the core substances and their chemical definitions or immutable definitions. That's a critical part to this. The main one that we're talking about today is the group 4 specified substances, and specifically the group 4 specified substance manufacturing information. The group 4 specified substance layer allows us to capture information that can get down into individual steps of manufacturing, as well as individual test methods and specifications that are used for a given substance in its context. In practice, we are only talking about the manufacturing elements of that. The specification elements of that are their own discussion, but for today, we have separated out, and in fact, in the GSRS software, we have separated out the manufacturing information, which again is really just the synthetic schemes, the synthetic diagrams. We have separated that out as their own elements to package together. So what does this mean? Well, KASA and GSRS, both of our teams worked closely together to try to take those elements that are specified in the ISO standard for Group 4 specified substance manufacturing and try to use them to systematically capture that information that's found in synthetic pathways submitted in these PDF forms during the review process. So the GSRS and KASA teams collaborated to capture this synthetic pathway information in the KASA platform by using a Group 4 specified substance manufacturing form that was developed in GSRS. So what this does is it enables assessors to capture the synthetic pathway and process details in a structured format, which is of critical need. And this form will be used in KASA for drug substance assessments for ANDAs, NDAs, and DMF submissions. So to jump into a little bit of what this means, we should consider the two ways that synthetic pathways can be entered. The current way is what we see on the left, which is as a picture or PDF. So these are non-machine readable, but often very pretty looking diagrams that describe the synthetic steps used in manufacturing a given substance, whether it's an intermediate or an API. It's a set of chemical reactions that you can see at a glance and get some understanding of the synthetic pathway. On the right, you see a screenshot from the machine-readable GSRS form that can enter the same information. On the right, the nice thing about this is users can enter individual elements, and they can update and change in a standardized way what reactions are done, under what conditions, and how each reaction flows into the next reaction in a machine-readable way, where this data can be databased. The important thing about databasing is, as informative as the diagram on the left is, it is still just a series of pixels. And a series of pixels is very difficult to search across. If a question arises about which reactions involve a certain solvent across the entire set of DMF submissions, this is a very difficult question to ask if the data we get remains as just the diagrams that we see on the left. Whereas if we can take that information and turn it into machine-readable forms that can be databased on the right, then these kinds of questions become trivial to ask. And that is our goal, is to make it so that it's very easy for this data to be indexed and standardized and searched across. So let me give some information about how this is done practically. What you see on the screen is a screenshot of a slightly out of date now, but mostly up to date synthetic pathway form, a Group 4 specified substance manufacturing form from GSRS. This form is machine readable, but it's also user friendly. Users can add information about each individual step of the process, and they can chain those together to fully define their synthesis. I'll just call attention quickly to the top. There are 3 elements that we use to show the synthetic pathways. The first is the form view, where information can be entered in a systematic way about each individual step and process. The second is the step view, which shows each chemical reaction one at a time. And the final is the scheme view, which shows a diagram very similar to the PDF and picture diagrams that we've seen on the previous slide, just made from the machine-readable form that's entered in the form view. This is how the form works. Currently, the set of operations works like this. First, you obtain the synthetic pathway. So for the reviewers at FDA, this means finding the section, finding those PDFs and those images that are contained in a submission. In GSRS more broadly, for folks who want to use the form outside of the DMF process, if you find any area where a synthetic pathway is described, if it's in a publication or a textbook or wherever it might be, you could use that instead to populate the form rather than something that comes from a DMF submission or NDA submission. Either way, once you've obtained the synthetic pathway, then you need to populate the form. Populating the form involves defining what the processes involved are and what the steps involved are. Each synthetic pathway has at least one process, and that process must have a set of steps. Now, each step is really just a chemical reaction. Sometimes it can be a purification step or crystallization, but in general you can map them to chemical reactions. So each step must have some starting materials, which are really input materials for that step. Reactants, you might call them as a chemist. Then there are solvents and reagents, which include catalysts as well. Those are the things that go over and under the arrow on a reaction diagram. And finally, there's resulting materials for that step. And those resulting materials don't have to be final resulting materials. They can be intermediates. They can be drug substance APIs. They can be intermediates used in the next step. They are just the results of that reaction. And finally, you can add process controls, things like temperature, time, pressure, conditions for that reaction that are important. So when you have a diagram like this, which is just a set of pixels again, it's just an image, you can take this information, realize that first there's one process. Typically, we define a process as a set of reactions that result in a material that will either be completely isolated or will be used in combination with some other material from another process to form a convergent synthesis. So here we have one process. We have the initial starting material. We have solvents and reagents for each step. And we have intermediates that are formed along the process. So we would be entering 1, 2, 3, 4 steps inside of one process, finding the relevant substances to map to for each one of these steps. So in practice, the form works like this. So here in the form view, the user would enter the first step, step 1 of 1. They then click Add Starting Material to add the initial material that's used in the first step. What I showed in the previous diagram, the very first step was had chlorobenzene as the first material entered, the starting material. The user can query by that green arrow by typing in the name of the substance, or the CAS number, or the UNI, or any number of other identifiers that might be used. And they can use this to find the unique identifier in GSRS for that material. And once they've done that, then that's present in the form. So here you can see that when typing in the name chlorobenzene, you get a list of drop-downs that allow you to see suggestions, and clicking on one of them reveals that chlorobenzene has this chemical structure, and you can decide to change or modify that selection as needed. But here you've selected it to be your starting material for this step. I should note that we also allow you to search by chemical structure. So you can click that search by structure button, which will pop up a little window that allows you to draw chemical structures and do searches based on similarity, or based on substructure, or based on exact chemical identity, to try to find the most appropriate starting material if you don't have a convenient name. So after you've entered each of these steps, you'll be able to see this simplified step view, which you see here on the screen. Each individual step, you can see what's on the left-hand side, the set of starting materials, and there can be more than one. In this case, we're only showing reactions that have one starting material. But you can see the set of starting materials. You can then see the reagents and solvents and catalysts over and under the arrow. And then finally, you can see the resulting material that was produced within that reaction step. In this case, most of these are intermediates. You can see that we make an intermediate here. The intermediate is used to produce another intermediate, which is used to produce another intermediate, which is finally used to produce the end product. So overall, the scheme view, that diagram, we can produce that final diagram in a simplified way from the machine readable form that's entered into the entry forms. And we can make a single image that still encodes this information. So we can take each individual chemical structure, we can say something about its name, we can identify the identifiers, the UNIIs that are used for each of those, and we can show either in a collapsed view or an expanded view the details about each of those steps in that scheme. And then we can store this scheme view as a picture in the database, as well as the machine readable forms. So a reasonable question is, why go through the trouble of entering this data into a machine readable form? Let me speak a little bit about some of the capabilities that become available when this data is entered in a machine readable form versus just an image of a diagram. We can ask questions, we can do queries that are like the following. We can say, let me find all routes that use a specific solvent. This has become very, very important for us to do as an agency over time. And having this data structured in a readily accessible database way is invaluable for answering questions like that. We also could find all the routes that involve a specific starting material or an intermediate, which has similar utility. We can also quickly compare similar pathways. Without having this data be machine readable, comparing similar pathways would typically involve pulling up 2 different PDF images and just visually comparing them. And while that works for small synthetic pathways, the larger they get, the harder it is to do this. And the choice of even how to draw a synthetic pathway is very dependent on the submitter. So having a structured way to deterministically create a diagram makes it easier to systematically and visually compare similar pathways. We can also do things like find all the routes that involve a substance with a specific chemical motif or substructure. And this is critical. We don't just store the images of these materials that are used. Each of these materials are backed by UNIIs. They're backed by full chemical definitions of the substances. And that makes it possible to ask very detailed, deep kinds of questions about those materials and find routes that involve them, find literature that's associated with them, link out to public databases that refer to these same materials. It can greatly accelerate the ability to ask complex questions. And this is also important. Having this data in a machine readable form allows us to do things like accept structured synthetic pathways directly. Currently, we can accept images that people give us of synthetic pathways, but we don't have a structured form yet for them to give them to us. But working with partners at NIH NCATS, for instance, they have been able to enter data they find from public sources about given synthetic schemes, and they can just fill out a form like this, export the JSON, and send it to us through an email, and then we have that same data ready to use in GSRS. So here's just some final thoughts. KASA and GSRS collaborated to develop a structured machine-readable G4 SSM form, a synthetic pathway form. This allows assessors to create and visualize the manufacturing process. This allows for advanced search capabilities across the synthetic schemes, the synthetic pathways, and it enables rapid comparison of synthetic schemes. G4 SSM form will be made publicly available for download in 2023. This is an open source public domain tool that allows us to make these diagrams. We are using it now internal to FDA, but the code is entirely open source, and you can go to the link down below to see some examples of this. It will be made publicly available for folks to download and use as they would like in 2023 though you can just click there now if you'd like, and you can still see the details of the source code even now. And finally, I'd like to give some thank yous to all of my colleagues here. Frank Switzer, Larry Callahan, Marlene did an amazing job putting together the slides here. Our GSRS developers, both at FDA and at NIH NCATS, have been phenomenal. And the KASA team has been just a pleasure to work with. And it's been just a very positive, productive, and collaborative project. I thank you for your time and your attention.
Thank you all for the great presentations. To our audience, please remember once again, if you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. In addition to our presenters, we'd like to welcome to the Q&A panel David Green, Senior Pharmaceutical Quality Assessor in the Division of Lifecycle API, Office of New Drug Products and OPQ. It looks like we have some questions coming in right now, and the first group of questions will be directed to Dr. Naomi Kruhlak. And here is the first question for Dr. Kruhlak. In the absence of experimental evidence on the preponderance of one tautomer under the conditions of an Ames test, does the agency perform the QSAR analysis on both possible tautomers of a given structure when the sponsor included only one?
Thank you for that question. Yes, we do consider other possible tautomers when performing QSAR analysis. Fortunately, some of the software tools that we use are actually able to calculate multiple tautomers for a structure and generate the predictions independently for each of those structures, and then we can use that information and apply expert knowledge to come to a conclusion for that substance. But in cases where the software doesn't automatically do that, our expert review process would certainly consider tautomerization and possible differences that we might see in the outcomes of the models or the outcome of the Ames assay due to those different structures. So it's definitely something that we consider.
Thank you for responding to that question. We do have a few more questions for Dr. Kruhlak, and here is the next question. Is the SD file an alternative to running a QSAR analysis for impurities?
Thank you for that. No, the short answer is no. It's not an alternative to a QSAR analysis. It's intended as a more efficient and accurate way to send impurity chemical structures to the agency for review. But a QSAR analysis is still needed for each known and potential impurities in accordance with ICH-M 7.
Thank you for responding to that question. We've got a couple more questions for Dr. Kruhlak, and here is the next question. You mentioned that counter ions and charges should be removed from the structure for QSAR analysis. Should they be included in the SD file?
The answer to that question is yes, they should be included. The SD file is intended to accurately represent the chemical structure, so that would include any counter ions or charges. A detailed list of formatting conventions was presented by Dr. Marlene Kim and is actually available in the SDF file quick guide. But if we do need to perform a QSAR analysis at the agency, we would do that minimal processing to remove those other counter ions in order for us to run it through our models.
Thank you for responding to that question. We've got a couple more SD file questions, and here is the next question for Dr. Kruhlak. Should QSAR predictions be included in the SD file itself?
Thank you for that one. No, we are requesting that the data in the SD file be limited to a small number of fields, and those are outlined in the SD file quick guide. So there will be a structure, and then the associated data fields will be name, application number, ID, role, Unicode, cast, and notes if needed. The QSAR predictions themselves should actually be presented in a tabular format in section S.3.2, where the name of each chemical in that table would then correspond to the name that's provided in the SD file. Thank you.
Thank you for responding to that question. We have one more question for Dr. Kruhlak on the SD file, and here is the question. Should the API structure be included in the SD file?
Thank you for that one. Yes, the API structure should be included. Ideally is either the first or the last chemical, so it's easy to find within the file. Keeping in mind that for QSAR analysis, predictions are generated for the API as well as the impurities. The API can serve as an experimentally tested comparator and facilitates the application of expert knowledge to the QSAR predictions. So for example, if an alert was present in an impurity, and it's also shared with the API in the same chemical environment, and that API is negative, we can use that prediction from the API to dismiss the alert in the impurity and assign the impurity to class 4. So we want to have the API there so we can run the impurities and the API all in one go if we need to.
Thank you for responding to that group of questions. Moving on to our next panelist, we have a few questions that came in for Dr. Frank Switzer. And here is the first question for Dr. Switzer. If the UNI number is not available for a given molecule, can this field be left blank in the SD file matrix?
The answer is UNIs are recommended, but not required. So yes, it can be left blank.
Thank you for responding to that question. We do have another question for Dr. Switzer, and here is the question. Is GSRS publicly available?
Available. The version behind the firewall is more extensive for investigational substances particularly. But approximately 135,000 UNIs and their descriptions are available primarily at a site at NIH's NCAT's center is the link is in my slides.
Thank you, Dr. Switzer, for responding to that question. We do have one more question here on GSRS. And here is the question. Can GSRS handle impurities that are presented as a group? For instance, an oligonucleotide, it's common to describe an impurity by the structural modification of a moiety in the API that can occur at any one of the nucleotides in the sequence or at a subset of the nucleotides.
For the work that requires QSAR, individual structures are needed. So even though they could be represented, it's not preferable to do that because each individual structure would need to be analyzed.
Thank you for responding to that question. Moving on to our next panelist. We've got a few questions that came in for Tyler Perea. And here's the first question for Mr. Perea. Is GSRS G4SSM for industry or FDA?
That is an excellent question. The answer is ultimately both. Currently, we are using this form internally as part of the KASA effort. but there's nothing to stop industry from using the form directly to help them organize their own data. The software is publicly available. It's open source, and we hope that people will use it to help organize things and give us some feedback. So it really is for both, but the primary use case right now is internal to FDA.
Thank you for responding to that question. We've got a few more questions for Mr. Perea, and here is the next question. When the structure of the starting material is not common, as is the chlorobenzene, which has been used in the example, does the system allow to draw more complex chemical structures?
That is an excellent question. The answer is yes. As Frank alluded to, the full GSRS set of features are behind the scenes on this form. So if you're attempting to either search or register for a complex substance, you have the full battery of tools that GSRS comes with. So that includes the ability to draw complex structures, to import mol files from ChemDraw or something, to resolve names based on IUPAC or other external identifiers, and even to import a image like a PNG or a PDF of a chemical structure and have it attempt to turn that into a machine readable form. So all of those things are features that are built into the form.
Thank you for responding to that question. We do have another question for Tyler Perea. And here is the question. And it's a two-part question. We'll have Mr. Perea respond first, go back on mute, and then we'll bring in Dr. Switzer to also respond. And here is the question. How does UNI tie into global harmonization among all other ID databases that are already in use or under development?
That's an excellent question. So I'll say a little bit about this, but I think Frank can say more about it. The UNI is the first identifier for substances that is built off of the concepts of ISO IDMP with the granularity required to distinguish substances. So there are many other very, very useful identification systems out there, obviously CAS being one of them, PubChem being another one, that are publicly available or publicly queryable. For those ones that, but they don't all meet the IDMP standards. For those ones that are used by other regulatory agencies, they've used the UNI to help bootstrap their compliance with IDMP. And so the UNI is used heavily in mapping exercises already to try to make sure that two things that people used to think were distinct really remain distinct, and that two things people used to think were the same remain the same after deeper consideration.
And also remember that the GSRS allows for description of substances of many types, not just small molecules, although the emphasis today has been about chemical structures. But the managing the molecular weights of polymers, managing monoclonal antibodies, managing plants and microorganisms are all part of the purview of GSRS and the UNI.
Thank you both for responding to that question. We have a couple more questions that just came in for Tyler Perea. And here is the next question. How long does it take to populate the form? Hello, we're having some connectivity with Tyler right now, some connectivity issues. Our tech team's going to reach out to Tyler, so we'll move back over to another one of
I'm
our.
back on. I apologize. I had bad internet connection just a sudden.
I'll repeat the question here, and here's the question for Tyler Perea. How long does it take to populate the form?
So that is an excellent question, and it really depends on a few factors, such as the complexity of the pathway. The steps that take the longest are actually just finding the substances that go in each step. But if you have the UNIIs or a CAS number or a name, it's quite simple to find them. And as the other question asker mentioned, if there's something more complex, how do you deal with that? There are still ways, if it's complex, as long as you get familiar with the tools to find based on names and on images, it can be pretty quick too. But I'd say once you get the hang of the form, as long as there's nothing that's really outstanding that's needed, it takes about 5 to 10 minutes to enter a 5-step process with all of its parameters. And so that's you'd probably expect somewhere in the order of 5 to 10 minutes for most pathways.
Thank you for responding to that question. Moving on to our next panelist, we have a few questions that came in for Dr. David Green, and here's the first question for Dr. Green. Besides the structure and Unicode, what other information is in the substance registry file?
Oh, thank you for that question. Someone who's used the GSRS website, the GSRS substance file contains all known chemical names, substance classes, various codes such as a UNII, BDNUM, CAS number, etc., molecular weight, formula, references, and much, much more. I encourage anyone interested in knowing what is in a GSRS substance file to explore the NIH GSRS NCAT's website. A link was provided on slide 14 of Frank's talk. That's very informative.
Thank you for responding to that question. We have another question that came in for Dr. Green, and here is the question. How helpful will the machine-readable synthetic pathways, a GSRS, be to the FDA review staff?
Thank you for that question. As someone who's used this powerful tool, I would say once the reviewers become very familiar with the form and how it's used, it will become extremely helpful for the FDA review staff. Especially for the first time, analytics can be performed on the pathway data. These tools will make it easier for reviewers to identify potential impurities and other possible review issues easier. Another step, I think, to increase our efficiency in our reviews. Thank you.
Thank you for responding to those questions. We just had a few more questions come in for Dr. Frank Switzer. And here's the first question for Dr. Switzer. How long does it take to receive a UNII?
If we receive the a good question, thank you. If we receive the information we need, anything that can lead us to a chemical structure for small molecules, I mean, typically we get the UNIIs back in about a day. More complicated things can take more time, and oftentimes those involve a dialogue back and forth between our team and the requester.
Thank you for responding to that question. We've got a couple more questions that came in for Dr. Switzer, and here's the next question. Will FDA protect confidential substance information?
Yes, the GSRS, great question. Yes, the GSRS is designed to be able to protect every piece of data selectively, and so we can, in many cases, we can release just the UNII and a name which is not confidential, and then release other data as it becomes public. Thank you.
Thank you for responding to that question. We have a couple more questions come in for Dr. Switzer, and here's the next question. Can the name associated with a UNI be different from the name used in the labeling?
Yes, good question. Yes, we often have the same compound that will have multiple names in multiple jurisdictions, multiple domains, and we try to use a single name associated with the UNI in the electronic listing, so that consumers, patients can read across different labels and find the same name for the same thing. But yes, we manage many names. Thank you.
Got one more question that just came in for Dr. Switzer, and here is the question. I searched for a UNI using a CAS registry number and found multiple matching UNIs. Which one should I use for the listing?
A good example of that kind of situation are polymers, where there'll be a single CAS registry number for the same polymer with different molecular weight ranges. And in those cases, you should use the UNI that best describes your particular ingredient. If it's polyethylene glycol 3350, you should use polyethylene glycol 3350. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we've got a couple questions that just came in for Tyler Perea. And here's the first question for Mr. Perea. Where can the public download the form, and does the form run on special software?
That's an excellent question. So the source code for the form is open source and available on GitHub. It will be packaged together with a future release of the full GSRS software that NIH NCATS distributes. The hyperlink to the code, the current version of the code, is on slide 18. That's just a small version of the form. There's more involved in it. But if you're interested in actually playing around with the source code, you certainly can by just using the link on slide 18. The software itself uses just a web browser for the form. So you don't need any special software to use the form, as long as someone has already set up the server. To set up the server locally requires some Java software installed.
Thank you for responding to that question. We've got just a couple minutes left. We're going to try to squeeze in one more question that just came in for Tyler Perea. And here is the question. When using the form outside of FDA, how do you enter a substance that does not exist in GSRS?
That is an excellent question. So users can request a UNI, as Frank got into, for a substance just by emailing the FDA SRS at fda.hhs.gov email address. A UNI can be requested at any time, and once the UNI is present, it can be entered into versions of this form that are backed by the UNIs. You can get a full local copy of the open source GSRS software also through our partners at NCATS. And if you installed that locally, you could even register your own substances locally for use in making these pathway diagrams.
Well, that's all the time we have for questions. We'd like to provide a huge thank you to all of our presenters and panelists for answering numerous questions that came in today. It's our pleasure to bring back Commander Skanke to provide some closing remarks. Welcome, Commander Skanke.
Thank you very much. Yeah, it's been a wonderful day, a great workshop, and I do have a lot of people that I'd like to thank for that. Captain Brenda Stoddard and the entire SBIA team. They did a wonderful job in the spring of 2021 when we did our 2-day DMF workshop. And they've again done a terrific job on this workshop. They help us every step of the way, and we couldn't do it without them. So, we extend our sincere thanks to them, and we look forward to working with them again on the next one. The planning committee, they did a wonderful job. We started planning the workshop 4 or 5 months ago. They performed a lot of work, both on the administrative aspects of putting on a workshop and the technical aspects of putting on a workshop. So to Yingzi Wang, Chin Shi, Fardak Abelwerde, Gianni Pereira, Barbara Scott, and Aaron Skoda, my sincere thanks for a wonderful job. I would like to thank the speakers. The content was wonderful. The answers to the questions were great. We did try to plan in this workshop to allow for a little extra time for the Q&A sessions. So there was more talking with you rather than just talking at you as far as a virtual format will allow. So, I do think that enhanced our workshop this time by allowing a little more time for questions. I hope you all appreciated that. I'd like to thank all of our participants. We have over 3,800 registrants from 80 countries. So again, another very well attended workshop. So thank you very much for your time. And I hope you've taken away information that you can use. As far as a sort of a common theme that permeated the entire workshop, I'd say that would be improving the efficiency and consistency of drug master file and drug substance review as we move forward. You heard in the morning about the DMF review enhancements and how those can increase the efficiency of the overall generic drug review process. In the afternoon, you heard of the amazing progress we've made in KASA and the structured data formats and infrastructure that's available to support our transition into KASA early next year. And I'd just like to point out one very important piece here is industry's participation. You've got a lot of information from this workshop and other sources that you can use. And we're only really going to be able to realize the full benefit of our procedural GDUFA 3 enhancements and KASA if industry takes the information that we're presenting here and puts it to use in your submission strategies and your submission formats so that we can realize the full benefit. So, for example, we anticipate that somewhere between 80 and 100 DMFs on an annual basis would be eligible for a DMF prior assessment. But we won't do 80 to 100 DMF prior assessments unless industry submits to us the proper requests. So, it's going to take some effort on your part to use this information and then take advantage of it. And the same thing goes with KASA. The tremendous benefits that KASA has to offer will only fully be realized if you take advantage of the structured data formats and as we move forward, the ability to submit synthetic schemes in a structured format. We won't realize the full benefit of that unless industry help us out and support us in that. So please take this information and support us as we try to move DMF and drug substance review to a more efficient place. That's my ask going forward. So again, with that, I'll close things out and thank you again for your attendance and take care until the next time.